All right, in this video, we're going to be looking at the structure and function of biological macromolecules and looking more so at how the directionality of these molecules changes the way that they function. We'll be looking at nucleic acids, proteins, and carbohydrates in particular. All right, so with nucleic acids first. Nucleic acids have a very distinct directionality, and this will come in, we'll talk about it now, but we will talk about it again, particularly when we get into genetics and talking about how the DNA molecule is situated. But you can see here, each one of these carbons, and when you see a ring structure like this, the carbons are just assumed rather than writing them all out. Anytime you have this kind of juncture, you can assume that there's a carbon there. Remember, we talked about how carbon has that ability to bind together many different things. And so here you have these carbons, and each one of them is numbered by these 1', 2', 3', 4', and 5' carbon. And so when we're talking about the sequence and the direction of a nucleic acid, we typically refer to them in the 3' and 5' sugars, or the 5' direction. And we would say that these molecules have this 3' group here and this 5' sugar that is up here by, or this 5' carbon, excuse me, that is up here by this phosphate group. And so this determines the direction that this molecule will go. All right. And we typically say that a DNA molecule runs from 5' to 3'. So you have your 5' carbon here, right? And you have your 3' carbon here that has this hydroxyl group on it. And notice here, it's the opposite. So you have 5', 3'. These two strands are parallel to one another, but they're going in opposite directions. And so what we say about the DNA molecule is that it is antiparallel. It is parallel, but they are going in opposite directions. Now, when new nucleotides are added to this DNA molecule, they are always added to the 3' end. And we'll talk more about that when we get to DNA replication. And so also a couple things else in DNA, notice how these two strands, you have this strand going this direction, this strand going towards the top, and you have the nucleotides that are always going to connect to each other. The thymine always connects to the adenine, and they do so with just 2 hydrogen bonds. All right, so it's a strong connection between them. Remember, hydrogen bonds are a connection between 2 polar molecules. So this NH2 here has some polarity. This oxygen has some polarity, and they get along really well. The same thing you see here between these 2 atoms, and they're connected to each other. But so the A and T have 2, but the C and the G, the guanine and the cytosine, they will connect to each other with 3 hydrogen bonds. And so a molecule that has a lot more C's and G's will tend to be a little bit more stable than one that has a lot of A's and T's. Should make sense to you, just because there's more bonds that are there. Those hydrogen bonds give that stability. And again, as we're looking at DNA, remember the linear sequence of DNA determines its biological information. And so any change to this sequence is going to change that information. All right. So if I had, instead of a T here, I had a C here, that would change this completely. It changes the code. DNA, most DNA sequences code for proteins. And so those proteins then would be changed. And remember, the shape of a protein determines its function. And so if you change the shape, it is no longer has the same function. And that could have bad consequences. And so here's another picture of DNA, and you can see the A's and T's and C's and G's and how important they all kind of line up. C's and G's with 3 hydrogen bonds, A's and T's with just 2, and they're going in an antiparallel thing here, directional. And notice all the A's and T's and C's and G's. And again, if you just take out its G here and put a T there, it's going to completely change the way that this molecule is read. It should be like taking words out of sentences or adding words to the middle of sentences, same sort of thing. All right, so when we look at proteins, proteins also have some directionality. We talked about the N-terminus or the amino terminus and the carboxyl terminus or C-terminus ends of individual amino acids. Proteins are comprised of linear chains of amino acids. And so it's never just one, it's a lot of different amino acids. And they always have this N-terminus and C-terminus ends. And so when these two ends join together, here's the N-terminus or the C-terminus of this one and the N-terminus of this other one, they join together with this peptide bond, which is a covalent bond, very strong bond. And so a chain of amino acids then there could be hundreds of these amino acids in an individual chain, and this sometimes we'll call that a polypeptide. And so you can see why we'll call it a polypeptide because, well, it's multiple peptide bonds. With proteins, there are 4 levels of structure that we need to be aware of here. The primary structure is what we just talked about, where you have an individual chain of amino acids, each one joined together by those strong peptide bonds. And so I like to think of this as like a string of Mardi Gras beads, right? It's just kind of all chained together. Not a lot of function there. In order to create some function, there would have to be some dimensionality to it. And that's what begins to happen with proteins because of those R groups. And so this group here is going to either do one of two things. It's going to start coiling up and forming what are called alpha helixes, or it's going to start folding up and it's going to make these pleats, and these are called beta pleated sheets or just beta sheets. And these sheets are forming hydrogen bonds with each other, kind of forming this structure here. And these structures, these helixes and these sheets will all start to kind of connect together as well. And this has to do with those R groups that are found in the amino acids. And so you have the helices that will stick to the sheets, that will stick to other helices and sheets. And then you have this tertiary structure start to form. And this tertiary structure is a three-dimensional structure. A lot of proteins at this point, they are done. All right. And there's lots of different ways that they can combine together. There can be covalent bonds, there can be ionic bonds, there can be hydrogen bonds, lots of different things. Sometimes you'll see one specific type of covalent bond mentioned as a disulfide bridge, which is a strong bond between two sulfur molecules. There's a couple of amino acids that have sulfur in them. And so this structure here is many times considered a completed protein, and it can go off and do the thing that it's supposed to do. Some proteins, though, have a 4th level of structure called a quaternary structure, and this is when you have multiple tertiary structures that are interacting together. Again, not all proteins exist at this quaternary structure, but a lot of them do. Some very important proteins, hemoglobin, which carries oxygen in our blood, for instance, is an example of a 4 sequence quaternary protein. And so it just depends on the structure and the function of the protein, whether or not it is going to have this tertiary structure or quaternary. But again, for most proteins, this tertiary structure is considered a finished 3-dimensional product that has a certain kind of job. And let's look at the directionality of carbohydrates. Then carbohydrates are comprised of linear chains of monomers. And so here you have these individual pieces of glucose, all right, these monomers, and this long chain here is a giant macromolecule or polymer. And notice here, depending on the way that they're bound up, it's going to change that structure dramatically. All right, so here you have cellulose. Cellulose is a building component for plants. Plants use them in their cell walls and in other things. And notice how they're bound together. You have these oxygens here. One goes in this direction up and the other one goes down, and they kind of trade off there. Whereas on the starch all goes one direction. They all combine this one direction. Well, this starch here isn't going to really want to be that close to another starch because of the way that this oxygen is situated, whereas one cellulose can line up pretty easily with another cellulose because of this every other formation. And this causes these two things to form different types of structures. So here's a picture of this. So here's a picture of starch, and starch, notice, has this branched kind of structure. Starch can't really get close to one another, so they've kind of make these branches almost like a tree. And glycogen has a similar kind of branched structure because glycogen is a very similar type of carbohydrate. And these two molecules have similar functions in the organisms that they're in. Starches can be found in plant products, and starches, the function of a starch is to store these glucose molecules for later use. Glycogen, very similar type of function in that it stores glucose, but except this is in animal cells. But notice they're both branched, and so they're good at this storing function. Whereas cellulose, notice, it's very orderly, all these nice straight lines. All right, so cellulose tends to form a structural type of material for plants. And this is also why when our bodies go to digest starch or glycogen, they can do so very easily because we have enzymes that are able to do this. However, we don't have enzymes that are good at digesting cellulose because of its more orderly and structured look here. It's more difficult to digest. And some animals have gotten really good at that digestion of cellulose, whereas others like us, it just kind of passes through us. But it does serve a important function in that regard as well. And so again, as we look at this, just note the structure of these molecules definitely determines its function and how they interact in their environments.